Hello, so I am recording now. Welcome, everybody. Thank you so much for joining us to the U24 Neurons Matter Network Seminar Series. My name is Victoria Oreira, one of the U24 Network PIs, and it's my pleasure to introduce you to today's speaker, Dr. Ishmael Abdus-Saboor, an associate professor of biological sciences at Columbia University's Zuckerman Mind and Brain and Behavior Institute, and a Howard Hughes Medical Institute Freeman Hrabowski Scholar. Dr. Abdus-Saboor's lab is really a pioneer in our understanding of those skin to brain axis, how sensory signals from the skin shape our experience of pain and social touch. And his team has developed also a lot of really innovative machine vision tools that allow us to measure pain responses at millisecond timescale. And his work on identifying touch neurons that drive dopaminergic reward pathways was featured in Cell and recognized when he was named one of the 50 scientists that inspire in the journal's 50th anniversary issue. So his contributions have been recognized with many honors, including the NIH Pioneer Award, the New Innovator Award, the McKnight Scholars Award, Pew Scholar Award, and Sloan Fellowship, among many others. And today he will be presenting on touch neuron populations that are necessary and sufficient for stress resilience. And I think is a topic that speaks directly to the exciting work that connects peripheral sensory neurons to central circuits that promote well-being, something that our U24 network really supports and values. So please join me in welcoming Dr. Ishmael Abdus-Saboor. Thank you.
Thanks. Wow, what an introduction. So what happened when your friends introduce you, right?
You're welcome.
All right.
No, we're so happy to have you here.
Yeah,
Take
let
it
me
away.
slides. Okay. Does this look good?
Looks
Okay,
good.
great. Yeah. So thanks for coming. I recognize some names in the amongst the attendees and happy to meet make new acquaintances and friends. Yeah, and thanks for coming back. I know we rescheduled this. We had this crazy snowstorm here a few weeks ago. We were just talking, some of that snow is still in the ground. It's pretty cold, but we're surviving. So my lab does a number of things, and what I'm really excited to focus on today is our work studying touch neuron populations and showing their importance, the necessity and sufficiency for stress resilience. And we do this in the mouse, and hopefully by the end of today's talk, you'll appreciate why we address these questions in the mouse. And I think this, as Vicky was just alluding to, there's exciting basic science discoveries to be made in this space, but I think there's also some therapeutic potential as well, and I'll try to convince you of that by the end of today's talk. All right, so to start very broadly on things that I think we all appreciate, social touch is absolutely critical for our happiness and well-being. If you just, do a Google search of your favorite animals, socially interacting or touching, you can find throughout the mammalian kingdom, right? Social touch is just widespread. It's important throughout development and parent-offspring interactions and for driving normal health throughout development. Even throughout life, it drives our bonds and our deepest relationships with friends and family. Touch is oftentimes at the center. As the focus of the second part of the talk, social touch can be quite comforting, as we all appreciate, can relieve anxiety and depression, acute and chronic stress. When we're down, one of our go-to strategies is to engage, hug, massage, touch, especially those who are closest to us. And during the absence of social touch, during periods of isolation from sickness or other things, COVID pandemic, and especially if this isolation is chronic, we can see all sorts of behavioral and brain deficits in humans and many animal models that are studied. So social touch is absolutely critical, and because of this, our nervous system is wired and tuned from detecting signals in the periphery to relaying that information to the spinal cord and brain so that we crave this sensation. But despite this ubiquitous nature of social touch, I think there's a lot left to discover, especially thinking about the integration between body and brain, filling in cellular molecular circuit details, and this is something that really fascinates us. Okay, just for by way of a few more moments of introduction before I get into our work. the what we do know about the initiation of this social touch circuit, which would be familiar to most people here, I assume. They're dedicated sensory neurons that appear very important for social touch. One class, these C low threshold mechanoreceptors, they're not the only ones, but they're been the most heavily studied in this context for numerous reasons. So, just by way of really quick background, you all probably know this paper where the cell type was first discovered in humans in 1990, where you could do psychophysics and electrophysiology, stick an electrode in a person's forearm, and then with this stroking cotton wool, and the person says this feels good, and you can see there are units that fire to this. A more recent sort of rendition of this you can see here again in humans. Beautiful. You can talk to people and they can tell you how things feel, and you can see these C low threshold mechanoreceptors activity towards this soft brushing. You can see the number of spikes to this slow stroking velocity across the skin, and people say this feels good. Something that I won't dwell on too long here, and this was beautiful work by colleagues of ours in the field, and including my postdoc mentor, Wen-Chin Liu. One thing that you see in this literature, which I think is interesting, that I don't think we discuss or have figured out, is that, or why these CLTMRs also respond to ramps, that are approaching noxious temperatures in both the heat and cool direction. The cool part we knew, but this paper showed also, there's responsiveness to heat. Is this a polymodal feature of CLTMRs, or it's important for the encoding? I think that's not so clear, but maybe a question of interest for the field. So this is in humans, and we have analogous populations, I would say, in the mouse, and I'll talk about these two today, mainly this first population. So this was work initiated by David Anderson's group at Caltech, and I remember reading these few papers when I was still a postdoc with Wen-Chin, and getting really excited. Thought these were just remarkable papers. So in this one, they just did neuroanatomy and just showed this, these sort of patchwork like anatomy in the mouse skin. This is the ear skin or back skin. Here they did calcium imaging and some behavior. And here, when they put the genetically encoded calcium indicator in these DRG neurons and did a hard preparation recording from the DRG terminals in the spinal cord, they could see calcium transients to this stroke. And also, they did chemogenetic body-wide activation and showed there was a place preference for this stimulation. And again, these results really excited me, and we decided to further expand upon these results, as I'll get into momentarily. The other population that is the CLTMR population in mice express TH, and is now a landmark paper in the field that I'm sure people are familiar with from David Ginty's group. You can see these TH positive DRG neurons responses to mechanical stimulation, and even similar to what I showed you in the human paper, responses to this ramp, this cold ramp. Now, in this population, this is Aziz Mokrik's lab has shown that TAFA4 is also a marker for these TH neurons in the DRG. You see like almost a one-to-one, 100% overlap in the DRG. This paper from the Ginty lab recently that people have probably seen, this wet dog shake paper, is a really beautiful paper showing you can stimulate these CLTMRs in the skin and get this really stereotyped behavior. Anyway, in this paper, they made a TAFA4 Cree-ER line, which I think will be valuable for the field because, as some of you may know, the TH line also hits the sympathetic ganglion, as shown here. But this TH mark, TAFA4 is not in the sympathetic ganglion, only in the DRG. So I think this will allow like specific DRG manipulations that can exclude the sympathetic ganglion, as they show in this paper. And David Ginty was kind enough to share these mice with us, and I'll show you some results we have from using this mouse line later in the talk. All right, so here's where we'll go today. So I'll talk about some of the work, as Vicky alluded to in the introduction. This first part I'll try to get through it relatively quickly. A mix of some published and unpublished work, this so-called skin, the spinal cord, the brain axis or pathway for rewarding touch. I think it sets up this part nicely, as you'll see, where we expand upon this work to look at these neuron types and stress resilience. And there will be a mix of published and some unpublished work in this first part, and then the second part is all unpublished and as you'll see, still ongoing. All right. So again, for the published study to this kick us off, this was led by my first PhD student, Leah, who's now a postdoc with Seth Blackshaw at Hopkins, and Bella and Will were technicians at the time, and now they've since stayed at Columbia, and they're PhD students in my lab. So we followed up on the Anderson paper, as I mentioned, with these B4 neurons, and for two reasons. One, although that paper showed that neurons could detect the stroking touch, they weren't implicated in any natural behaviors. And as Vicky mentioned, my lab likes to study behavior really intensely and develop new quantitative approaches to measure behavior. So we thought we could find some natural behaviors these neurons were involved in. And more importantly, there were no connections made to central circuits in the spinal cord and brain. And we thought this would be a good population to study to try to get at the skin brain connectivity. All right, so our point of departure was to just put channel rhodopsin, the blue light sensitive ion channel, in these neurons to activate them in vivo. And you can just see this opsin has an EYP fluorescent tag, and we can see associated with hair follicles in the mouse hairy skin. I'll show you unpublished results. So in this paper, we spent a lot of our time like just following the mouse around with the blue laser light through and shining light directly through the shaven hairy skin. But more recently, we've turned to a more robust approach so we're kind of converting away from that approach using this wireless optogenetic device. This device comes from this company, Neurolux. They have a number of probes. This is the probe that we like to use. We just implant this directly under the skin, and then you just need to have these antennas like encasing the whatever the behavioral setup that you're using. And here you can just see a mouse with this wireless implant. Um, See the mouse is perfectly happy, and you can see the implant flashing. This allows us to do these experiments remotely. We can test many animals at once, and it's just more robust. All right, so we can see that so during this wireless opto approach, we can see that day one we get a real-time place preference for the light. You see wild type animals, they go back and forth, but these animals, just one pairing, they prefer this stimulation. This is different than our paper where it took us like 5 or 6 days of conditioning. So this is just more robust. And with this wireless approach, this allows us to, I think, brain recordings are a little bit easier now and other sorts of behavioral apparatuses, including this operant assay. These again are still ongoing, and we're still kind of playing around with this protocol, but to show you early results where we can pair this with a nose poke, and we can see the animals really nose poke like crazy for this B4 neuron stimulation with this setup. And results like this are really convincing that this stimulation is inherently rewarding, as you would see similar things with like, sucrose or drugs of abuse, for example. Okay. All right. So this is a new stimulation protocol we've been using to stimulate these neurons. We're slowly converting over to this from the old approach. All right, so back to the published study. What about behavior? So we decided to focus on mating behavior. Clearly, there's a lot of touch that occurs during mating. It's a relatively easy behavior to score. It's robust readouts, and many people study it. So we decided to look here, and here was just a paradigm we used. We overreactomized the animals, and then we controlled when the females were in a state of behavioral estrus. And we did this over the course of multiple weeks, as others have done in this field. And what we see is that in wild type females, sexual receptivity goes up over time. But when we've ablated these B4 neurons genetically with this DTA approach, by the second and third week, many of the animals are no longer receptive. And they actually become quite combative, but only when the male touches them and attempts to mount. Okay, so these neurons this is at least one behavior that requires social touch behavior that requires these neurons. And there was also some beautiful work from Alex Chesler's lab and David Ginty's lab looking at other touch populations, the piezo in Chesler's paper, or the A beta mechanoreceptors in David's paper, showing those neurons important for sexual behavior. All right, so here we just asked a sufficiency question and we looked at dopamine dynamics and just as a canonical readout for reward circuitry. So to measure dopamine, we just used to grab the dopamine sensor and record it via fiber photometry in the nucleus accumbens. And I'm showing you this because we got a positive result where we shine blue laser light to the back skin and we see this dopamine release to blue laser light, but not controlled green laser. We like green laser because the animals can see it just like blue light, but it won't turn on channelopsin. Or if we activated the neurons in the anogenital skin where these neurons reside, we see this dopamine release as shown here. And again, this is the published result. We were using, I think, Grabda 1.0. We've since now moved to their third generation of Grabda. It's more robust and brighter, and the signals look even better. So I think this is a convincing result. And this finally, when we went back to the mating assays and measured dopamine release in female mice, you can see if we align the transients to male mounting, we get this nice dopamine release, but this is lost in the before ablated animals. This providing some dopaminergic sort of explanation as to why the females don't engage in that sexual receptivity. All right, so I tried to blow through that relatively quickly. There's more data if you're interested in that paper. We had a really key collaboration with Vicky's lab. We couldn't have published this paper without her. Her lab, as you all know, experts in many things, including spinal cord slice physiology and a really sophisticated mouse genetics using interrograde and retrograde viral tracing, allowing us to kind of fill in some of the intervening molecular details to some of the work I just showed you. And with that, we came up with this, can show that there is a circuit from B4 neurons to GPR83 neurons, these spinal projection neurons, following up on some beautiful work from Sebastian Choi and David Ginty. I think I said Ginty's name like 10 times already, but rightfully so. And from the brainstem to the VTA NAC circuitry. So about 3 to 4 cells seem to be able to get you from the skin to reward center for this rewarding touch. And Rui's lab taught us how to do these endoscopic single cell dopamine recordings that I didn't show, which we do a lot now these days. So we're really excited about this pathway, and I think it opens up many directions. This is just sort of one route. Social touch engages many areas throughout the brain. Animal has to keep track of internal state, the sex of the other animal, where in the body you're touched. So clearly there have to be other ascending pathways, and we're looking at some of that work now. I think I saw Maham on the call. It's been cool to see other groups kind of seeing similar things here in this paper from a number of friends of ours in the field. They activated the B4 neurons in the spinal cord terminals, so not directly through the skin. They see this really in both males and females, a really strong real-time place preference. This work really inspired us to move toward the wireless Neurolux optogenetics and get under the skin. So I think you just get better activation of the neurons when you don't have to take the blue light through the skin. All right. And this paper that came out at the end of last year, I thought was interesting. I'll just share briefly from a group in China. They did this paradigm, which a number of labs, including our lab and Vicky's lab and others, think is cool, where you can kind of see the cross species benefits of social touch. So a human experimenter can brush the animal, and the animals will prefer this stimulation. And as shown here, the animals, you can condition them to this brush, and they prefer those environments. But here, when they genetically ablated B4 neurons with this caspase-based approach, then this preference for this brush is lost. Okay? This is consistent with some of the things we've been showing you. And then this paper that was led by this more kind of follow-up from the study I showed you, led by Catherine Dulock's lab, thought this was a really nice paradigm that they have, where they looked at animals that were group housed, and then they did these acute isolations for 1 or 3 or 5 days, and then put the animals back together, and they saw a really remarkable behavioral finding. And that, depending upon how long the animals were separated, when they came back together, this would drive a heightened sort of reunion period. And Ding and Catherine noticed that as the animals came back together, they appeared to be engaging in lots of social touch. This paper is not really about the periphery. They really keyed in on 2 hypothalamic cell types that kind of sense this separation versus reunion state. And I think a cool idea from this paper is that, social touch is might be a homeostatic need state, just like hunger and thirst. When you're deprived of hunger and thirst, when you are have the ability to engage again, you kind of increase to get back to a set point. Perhaps something is similar with social touch. If you reduced it, when you have the ability, you kind of overcompensating to get back to a set point. But anyway, Catherine got in touch with us from seeing some of our results and thought the B4 neurons could be involved. So we sent mice and began to collaborate, same B4 DTA strategy. And here you can see that after this reunion, from a 3-day isolation, it drives this social rebound behavior, but this is greatly reduced in the absence of these B4 neurons, not down to zero. So other populations are clearly involved, and Catherine worked with David Ginty and Sebastian, I believe, and saw even greater reduction, knocking out the NAV 1.8 neurons, I believe it was. So I think this is cool for us to see yet another context, social touch behavior that requires these B4 neurons. All right, so what I've mentioned so far is that these neurons are important for this rewarding social touch through the skin-brain sort of pathway. How many behaviors rely on this pathway? Social touch behaviors. We've talked about mating. I just showed you this social rebound. We saw some work at this year's SFN in San Diego from Gloria Choi's lab at MIT. They had some really nice work showing that in a sickness model that as animals, if animals are allowed to socially engage, this social buffering could actually speed up the rate of recovery of sickness. And this appears to be dependent, and B4 neurons are involved. Just stimulating the B4 neurons could actually speed up their rate of recovery. It's really exciting work from her lab. And we're looking at other behaviors as well. And as I kind of alluded to, what are the spinal and brain circuits downstream of these sensory neuron inputs? We just looked at kind of one pathway from the parabrachial nucleus to the VTA NAC, but must be a brain-wide response. So we're taking a number of approaches. This will, work for another talk, perhaps. I will show you some of the CFOS mapping we've done downstream of this B4 stimulation in this coming part of the talk. We're doing opto fMRI. I won't show that today. And most recently, we're starting to do large-scale physiology recordings with neural pixels as we do natural touch and also opto stimulation of these neurons in the skin to kind of get at brain-wide networks. All right, so in the remaining part of my talk, I want to key in on stress resilience. All right, so social touch is a powerful stress reliever. I don't think I need to spend any extended amount of time convincing you all that that's true. Again, when we're acutely stressed or even chronically stressed, we all know the power of a mother's hug, right, or a loved one or friend. And but despite this kind of powerful approach that we have right in front of us, the molecular mechanisms for how touch is doing this remain largely unknown. Okay, and we would like to kind of begin to tackle this question. I think we have some of the tools and approaches to go after this. Okay, this is just a review that I think is really nice. It just came out a week ago, and the authors here in Switzerland made a really strong case of looking at this affiliative touch as a way to mitigate social stress, and where to look. This review kind of makes the point that if you look across the literature and mice and humans and mammals, some of the networks and systems that are involved in affiliative touch are also implicated in social stress. So, leading us to a blueprint of where to look as we kind of start to fill in these molecular details. And just one point from the paper that I think is just well stated, I just want to mention, the authors state, we conclude that despite the well-known buffering effects of social touch for stress and anxiety, more research is needed to establish somatosensory approaches, such as touch-based interventions, as a standard avenue in the treatment of stress-related symptomology. We argue that social touch, in fact, could be one of the most effective antidotes in the aftermath of social stress. I couldn't have said it any better. That's why I quoted it. All right. So, can one even begin to address this? Is this a tractable problem in a mouse? And I think so. And really beautiful work from Weijia Hung's lab, I think, demonstrates this nicely. This phenomenon that you see across the mammalian kingdom. When a member of your species is stressed, a companion animal can provide relief through touch. So, what they show in this paper, and this is just showing you the results of a foot shock stressor in mice, but they did a number of stressors in this paper. Take two animals together, stress one of them, put them back together. The companion animal can detect the stress on their friend or their cage mate and engage in this allo-grooming behavior. Okay, and they showed in this paper is at least in mice, it's largely driven by olfactory cues. They can kind of smell that state. And here's just some videos to show you a video of this affiliative social touch behavior that this is kind of one example. Hopefully you can see this mouse kind of on top of its friend, doing a lot of touching and grooming. Okay. I think there's another example coming. Right. Can detect that state and, really grooming and petting the other animal. Similar things you see in non-human primates and many other animals studied. And they showed that this behavior actually is meaningful for the animal. It reduces the stress and anxiety like deficits in this animal after they've gone through the stressor. Okay. So for Wage's lab, they're interested in the neural circuits that drive this animal to want to groom its friend. And they kind of, these papers that they publish map the circuits in the brain. My lab, we're interested in the animal that's being groomed. Like, why is it working? What are the circuits that are affecting it? So we're looking at kind of opposite sides of the same coin, but kind of related behaviors. All right. So the work I'll show you now from our group was initiated by Melanie Schaffler, a former PhD student. She's now in Denmark at Novo Nordisk, and it's been followed up and extended by two amazing new postdocs in the lab, Sasha Fulton and Anna Zukowskaya. Most of what I'll show you today has actually been generated by Sasha. All right. So we begin back to those before neurons that I've talked about thus far. So one of the first questions, and this is Melanie initiating the project, what are the functional consequences for animals grown up without these neurons using this genetic strategy? Looked at a number of behaviors. I'll just show you just some of the affective behaviors. So the sucrose, for people who aren't familiar with these tests, the sucrose splash test, you just kind of splash, and these are very common in this behavioral neuropsychopharmacology field. The sucrose splash test, you just kind of display a splash sucrose on the animal's back. Animals normally have a drive to want to clean themselves, but animals that have like a anhedonia, negative sort of state, they have like this kind of helplessness model, and they let themselves go and they don't clean themselves. For a swim, animal in a bucket of cold water, do they want to swim away or they just sit there kind of helpless? Open field is like a good anxiety-like test. Mice are exploratory, but if they're anxious, they don't want to go to the center. Long story short, we don't see any deficit just ablating these neurons at baseline. We couldn't find almost any deficit, which was surprising to us at first. But then we decide to push the animals a little bit with a subthreshold variable stress. This was shown by Scott Russo's lab at Mount Sinai to be able to be a state to kind of sensitize the animal for future stressors, kind of like almost a two-hit model. So when we do this 3 days of this variable subthreshold variable stressor and then go back and do those tests, we do start to see phenotypes. Not in sucrose preference, but that sucrose splash test I just talked about, for swim, reduced time exploring the center in the open field. So these are all hallmarks of depression-like and anxiety-like behaviors. The elevated plus maze, we recently done this. We need to do more, but less time exploring the open arms. In this context, we didn't see a social deficit. We do see deficits in court. This is interesting. With the court, we see earlier, 30 minutes after this 3-day stressor, we see court levels rise, this stress hormone to this level. Interestingly, if we look at the before ablated mice, even before they go through the stressor, they seem to have elevated court levels, suggesting that there might be even some stress susceptibility even before they go through the stressor. I'll come back to this point as well. So with these data, we can say that these neurons are required for animals to mount a normal response to a stressful scenario. And if they don't have these neurons, then the effects of stress are exacerbated. Okay. So that was the genetic ablation. We wanted to ask the question, well, is there like a real-time role for these neurons, or is this the neural circuits just altered so much with the animals not having these neurons throughout life? So here we turn to an inhibitory dread approach to silence these neurons, and inspired by the Weijia Hung's results, we did this. Approach where we did this acute restraint stress and then group housed the animals for 30 minutes. So as Weijia's lab showed, and we've been able to recapitulate in our own hands, that after the animals go through an acute stressor, the other animals can detect this in the cage and they do a lot of grooming to that animal that kind of mitigates the effects of stress. So we asked, if we just silence these B4 neurons during this grooming period, does this alleviate the positive benefits of this touch buffering? And the answer is yes. The before silencing those neurons removes the benefits of the touch, as you can see here. And this is this anxiety measurement of time in the center. And you see the animals do not explore the center. They have a very strong anxiety phenotype, showing that they're not benefiting from this extra touch that they are receiving from their cage mate. So these neurons are required not just through developmental, but in real time. Okay, so TAFA4, as I mentioned, this line we got from Ginty's lab. This is an inducible Cree, so we ablated these neurons at P14. And similar to the B4 result at baseline, we didn't see any deficits, but when we gave this 3-day variable stressor and then went back, this showing you some of the phenotypes, but they look almost very similar to the B4 DTA knockout, where you can see this strong anxiety phenotype after the stressor and the animals not wanting to go to the center of the cage as compared to the littermate controls, okay? Suggesting these neurons are also required to mount a normal response to a stressor. And we're going back now. I was hoping to have more results by this talk, but we're trying to do like almost everything we've done in the B4 line with this TAFA4 Cree line, and those are still ongoing. All right, so are there molecular and brain activity signatures of stress susceptibility in these B4 DTA ablated mice? So now I'll show you some molecular and brain activity data that would suggest that that's true as well. I just started with the behavior, now on to some other signatures in the knockout animals. So for molecular signatures, Sasha decided to look in the nucleus accumbens, an area that's been well studied for stress susceptibility. So we had some ideas of what sorts of things to look for if there were to be signatures. So with this single nuke RNA-seq, Sasha could resolve all the known cell types we expect to see in the nucleus accumbens. And then we've been going in and looking within cell types. Here's one result that we thought is interesting. If we just look in the astrocytes, astrocytes in the NAC upregulate a number of immune-related genes as and to mount stress responses, and we see even at baseline, this is all pre-stress. We see this signature indicative of a stress state in these knockout animals. If we another cell cluster we looked at within the NAC in these ablated animals was the dopamine clusters, and in particular the DRD2 cluster. And we can see reduced expression of the DRD2 high affinity dopamine receptor. But it wasn't just the receptor. If we look at dopamine response genes, and these are we got this list from Jeremy Day's group. These are genes that tend to be activated in dopamine neurons. We see like this whole network seems to be downregulated. So it looks like without these B4 neurons, this really alters setting up these canonical reward circuits. And this is again a hallmark of stress susceptibility in these models. Given the dopamine responses that I just showed you, we just decided to look to see if there might be generic deficits in dopamine-based reward learning. So in we just did this simple associative learning, pairing a light with a cue with the reward, and we can see that there are deficits in learning this task in the B4 DTA animals, and if we reverse the task as well. And if we just combine this with dopamine recordings, we can see wild type animals as they get these rewards. We see this nice dopamine, but this is lost in this B4 DTA mouse as shown here. We've repeated this with TAFA4 mice, DTA mice, and the results look nearly identical. And we think, again, these reward deficits are consistent with animals that have stress susceptibility phenotypes. We think they are related. All right, so the last sort of brain activity signature I'll show you before moving to gain of function experiments is this one. So it's been a fun collaboration with Rainbow Holtman's lab. She's at University of Iowa. This is her main postdoc paper when she was still at Duke with Cobb Zarasa. And anyway, they use this multi-site LFP extracellular recording from 7 to now upwards of 15 brain areas at once, and trying to get almost like an fMRI based kind of a functional connectivity map. And the really exciting thing about this work is that these signals are predictive. So they use this, people in the stress field will know, if you do any stress paradigm, for example, chronic social defeat stress, which is amongst the most common, you always have animals that are very susceptible or vulnerable, or some that don't seem to care, even though they've gone through the stressor. Similar to humans, some people are very susceptible to stress and others are quite resilient. And the mechanisms are still kind of unclear. The exciting thing about this paper is that they could find brain connectivity patterns that allowed them to predict which group animals would be in even before the behavioral results. So we thought this would be exciting to combine with our B4 knockout mice. Could even at baseline conditions, could we detect a stress vulnerability signature, kind of following up on what they show in this paper. In this paper, they validated this with a number of stress models, even some genetic models that where there's a propensity for stress vulnerability. So a lot of validation of these electrical functional maps. So anyway, we collaborated and sent a bunch of mice, and they did recordings. And this was led by Micah Johnson, a former PhD student in Rainbow's lab at Iowa. And they did these multi-site recordings and control and knockout mice before and after stress, okay, and recording across 7 different areas. This is real data from this experiment, and to give you a kind of a flavor of how the data looks. And there's a lot of math and machine learning that goes into the final output, the so-called electome factors. And in this 2018 paper, they found electome factors that, again, were predictive functional maps of stress susceptibility. All right, so this showing you this one of the results. There's a lot of data that came out of this, but this showing you one of the really nice results, I would say. During these recordings, before knockout mice, going through the same sub-threshold stressor I've showed you before. And I think this is nice. So this EF2 electome, this is one of their verified electomes from their prior and ongoing work. So activity here means stress susceptibility. So in control animals, you can see after they go through this stress paradigm and then are put in the cage to forced to socially interact with another animal. This is a stressful scenario, and we see this EF2 go up. Interestingly, I think excitingly, the B4 DTA mice have this EF2 network signature that is even reminiscent to a control mouse after they've gone through stress. Again, suggesting that even at baseline, there's a brain network structure that looks like stress vulnerability. And after the stressor, it gets even more exacerbated. Okay. So I think really strong evidence of this stress susceptibility. And here are the brain regions that seem to be driving this behavior. Okay, so everything I've showed you thus far in this part of the talk has been with knockout or silencing these neurons. And I think we are convinced that these neurons are required for mounting a normal response to stress, and animals have the stress susceptibility phenotype. So then we've started to be interested in the opposite side of the coin. What about activation of these neurons? How might this affect stress? So one thing we did just to kick this off is we looked at, well, does turning on these neurons engage a brain-wide activity structure network? I mean, we knew the answer would be yes, but what sorts of areas? So we just did this activating dread approach, and we worked with this company, Life Canvas Tech, to do this CFOS brain-wide mapping. We did experiments and then shipped them the brains, blind to genotype and treatment group, and they did their protocol and sent us back the data, and we can plug in the genotypes and treatment groups. And we saw obviously a number of things. I'm just highlighting some for the sake of shortness in this talk. Sanity check, we see the somatosensory cortex lights up as shown here and quantified there. We are turning on touch neurons, and the VTA is activated consistent with turning on reward circuits or social circuits. And we see decreased activity in areas that are normally have heightened activity after stressful scenarios like the prelimbic area or the lateral orbital frontal cortex. So this is reassuring and convincing us that perhaps this stimulation is working to kind of reduce stress areas. So then I think excitingly, still ongoing, we've turned to behavior, and I'll show you just two different behaviors. So we decided to do this. We wanted a paradigm where we know the animals will be stressed, and then we can go back and see, does this B4 stimulation alter the course of behaviors typically seen after these stressful periods? So we adopted this 7-day chronic variable stressor, and many people in this field use this test, and it's very stressful to the animals because each day there's a different stressor, so they can't kind of habituate. And people have shown at the end of this paradigm, there are many behavioral deficits and brain signatures showing that the animals are clearly stressed. Okay. We decided our intervention would just be this is using the external laser light, and at the end of each stressor, to stimulate these neurons for a few minutes. Okay. And then at the final day, we do behavioral tests. And excitingly, we're seeing some rescue of deficits, and some appear to maybe even have some some sex-specific rescues. So we look at anxiety-like behaviors that are induced after this paradigm, such as, again, reduced time to explore. So reduced time in the center, we see we can rescue this deficit by this stimulation. It appears to be male-specific in this context. Okay, so an anti-anxiety phenotype. If we look at sociality in this, at least in this paradigm, it seems like female mice are more sensitive. So this is time spent in a social interaction zone. So this is looking at a social interaction. Normally at the end of this 7 days, animals are quite antisocial and don't want to engage, but we can rescue this antisocial deficit with this stimulation. And interestingly, this is actually a dread experiment, but here's baseline and post-stress. We can see we can actually reduce cort levels, this stress hormone. And I'll come back to this result in a second because this is convincing. This has given us an entry point to think about, how might this stress rescue be working? Okay. All right. We've done this other approach too, and this is, as I mentioned, we're trying to convert all our experiments over to this wireless optogenetics because it's just more robust. We can do it remotely. So here is us using the wireless optogenetic stimulation. So here we're doing this chronic social defeat. So this is probably the most extreme form of a stress model in the rodent literature. So if for people who are not familiar, it's 10 days, and the C57 mouse is put into the cage of a larger CD1 mouse every day for 10 days. It's kind of getting beat up for 10 days straight. And at the end of those 10 days, the animals have these long-lasting signatures associated with chronic stress. Our intervention was to do 10 minutes of this wireless optogenetic stimulation at the end of each stressor, each day of stress. So, firstly, as this to kind of as a proof of principle of the system, we see at the end of these 10 days, we just do a real-time place preference to get an idea of how they're perceiving the stimulation. Animals that have not gone through a stressor, you can see, still have this real-time preference for the stimulation, consistent with work I've shown you before. But I think importantly for this assay, we see that this real-time preference for this stimulation is maintained after the stressor, which I think is important to in this context because one concern we had is that if the animals are going through this physical stressor and then we're activating these touch neurons, perhaps they would associate this touch with this stressor, and now it may become aversive. But does not appear to be the case. It still seems to maintain its inherent rewarding quality, as shown here. All right, so what about behavioral phenotypes? We see that in males now we can rescue this social deficit. So you can see here's the scenario. Normally, after mice have undergone this stressful period and are paired with a new CD1 mouse, they have this very, they don't want to be around this other mouse. They've just been, beat up for 10 days and really stressed. They spend a lot of time in the corner, kind of hiding, and we can rescue those deficits, with this stimulation. I think this is really quite exciting about the, potential therapeutic the potential sort of therapeutic implications of activating touch neurons to rescue, these chronic stress deficits. All right, in my last few moments, this is, really preliminary and ongoing, but where to start thinking in the brain for a mechanism that could explain some of these touch-based sort of inhibitory effects on stress. So I showed you before that we seem to be reducing cort levels if we measure cort in the blood after stimulating before neurons post-stress. So for those of you who don't know or not, I just need a refresher. The cort pathway goes through the so-called hypothalamus pituitary adrenal HPA axis to activate to release cort into the blood. It's initiated by these CRH neurons in the paraventricular nucleus in the hypothalamus. Okay, so we're keying in on these neurons, these CRH neurons. So we got a CRH GCamp virus, and we put it in the PVN, and just to validate this tool, we foot shock mice, and you can see nice activity of CRH neurons after animals have gone through the stressor, which many people have shown. Okay, so our paradigm, we went back to this paradigm that I just showed you, okay, the 7-day stress paradigm. End of each day, we're getting this before laser stimulation. And then on days 5 through 7, Sasha, heroically did these, it's a lot going on in this animal, but heroically did these dual recordings where she's recording CRH in the PVN and also dopamine release in the NAC. And from our early days, it looks like things are moving in ways we would expect. So here I'm just plotting area under the curve. And for these days where she did these dual recordings, we can see reduced activity in these CRH neurons and coincident increased activity in the grab the dopamine neurons or dopamine release. So perhaps part of the mechanism is for the stress mediated relief is is before neurons or touch directly silencing these CRH PVN neurons, and we're hard at work, further exploring this. All right, so just to wind down for today, in the first part, I've talked about the pathway we identified, the skin-brain pathway, important for social touch in mice. And it's exciting to think about, could we identify relevant pathways in humans? The human equivalent of TH or TAFA4 neurons or B4 neurons or even other, genetic types in the mice. And are there specific ways to activate those neurons? It could be a viable path for some therapies. And I think it's exciting to think about and explore. The second part of the talk, I told you, we can get these bidirectional effects on stress. Without these neurons, animals are more prone to have more stress or negative effects of stress. And activating the neurons, we seem to be able to reduce the stress deficits. So we kind of have the phenomenology, and the piece of the puzzle that we need to figure out is how's this all working. And I again kind of tease you. We think that the CRH neurons are going to be very important in this pathway, and we're hard at work trying to figure this out and conclude this. All right, so together, I think stimulating these social touch pathways may serve as a therapeutic that can mimic some of the positive benefits of social touch. And again, this was highlighted really nicely if you have time to check out this new review. Okay, with that, I'll end. Just want to thank you all for your attention. Amazing folks in my lab, our funding sources, and happy to take any questions.
Thank you so much. That was a wonderful talk as always. And right now, what we'd like to do is take any questions from the audience. You can unmute yourself, or you can also raise your hand. Although I can't tell the difference between the clapping hand and the raising hand. Hold on. Uh, the virtual applause. If you could raise your hand, I have one hand raised. Go ahead and unmute.
Hi, I don't know if
Hey, Kara, how's it going?
Hey, it was great to see all this amazing new data.
Thanks.
I was really impressed by the new implanted opto results and how strong that is. And you mentioned this like spatial
Hmm.
importance of touch matters, right? We feel different if
Yeah.
the touch comes in different areas, and I'm just wondering if you've played around with that.
That's a wonderful question. We have not played around with it, so just the short answer. At first we thought like maybe this wouldn't work because one maybe benefit of the laser is that we can kind of like move it around and kind of,
Mhm.
but with this implant, we're really only hitting like one area, and we thought maybe this won't even work, but
Yeah.
the results are even stronger. I think, it takes like 5 minutes to do these. So I think someone in the lab or in the field should we can play around with this, moving these probes, right, and look at the
Yeah.
responsiveness. I think that would be a cool project.
Yeah, it's amazing. I agree. This tiny stimulation area does this. It's really incredible. So
Yeah.
beautiful.
Yeah. Yeah. Thanks. Thanks.
Thank you for that question. Anybody else?
Go ahead, Francis.
Hi, Ishmael. We met a while ago, I think, in San Antonio.
Oh, yes, we sure did. Yep. How's it going?
I've got some really nice translational data. We published a paper in 2022,
Mm-hmm.
a study I did in Chile, where we basically stroke rats every day for 3 or 4 minutes at a
Mm-hmm.
slow speed or a fast speed. So slow group, fast group, and a control group. For every day for 2 weeks, they were subjected to a chronic, unpredictable mild stress paradigm.
Yes.
The story is at the end of that 2 weeks, a behavioral test and corticosterone test shows that those rats that had slow touch were completely resilient to stress.
Mm-hmm. Yeah, I must be
Maybe you've
familiar
come
with that
across that
paper,
paper.
actually. Yeah.
So again,
I'm familiar with that paper. Yes, yes, yes.
so that gives a clear indication that there's something. What has struck me about this, we then translated that in a study I ran in Russia. I took my name off it for political bloody reasons, but we
Mm-hmm.
looked at cortisol levels in family reared children
Mm-hmm.
and cortisol levels in institutionalized and a group of fostered kids. The institutionalized and fostered children's cortisol levels were up through the roof. And again, all we did for 2 weeks was use this wonderful peer-to-peer touch program where basically these kids are just gently touched every day. After
Mm-hmm.
2 weeks, those cortisol levels are down at the same level as the family reared children. Now that I think is supporting exactly what you're
Mm-hmm.
aiming at here. Is this neurofiber didn't evolve for no bloody reason. I've called it vitamin T. It's a fundamental necessity for regulating stress systems. In the early days, we thought these CTs were pleasure nerves, but I think we're more now of the opinion that, as you are agreeing with, that this is stress regulation. And it's the less we touch, the worse it's going to get. So I think it's absolutely fundamental that we get these translational models out of the door, Ishmael, with your phenomenal work that you do with these mice. It's unbelievable. But
Oh,
it's
thank
got
you.
to
Thank you.
translate.
Yeah, it's very kind. Yeah. One thing I'll add too, I didn't get to plug in too. There's, a really beautiful paper in bioarchive now from Vicky's lab that we work with and a number of people probably here showing the oxytocin, circuits in the hypothalamus downstream of social touch. And, we need to show this data. Maybe we need to work on this together, Vicky, as we always tend to do. But we think that like maybe we're going through because people have shown that oxytocin neurons can directly inhibit these CRH stress neurons in the PVN. So maybe that's part of the circuit through oxytocin to direct inhibition of these stress driving CRH neurons. It's one of the things on our long to-do list to try to figure out.
Karina, go ahead.
Hi, Ishmael. Thank you. Thank you for your
Mhm.
talk. It's really impressive data.
Thank you.
As you mentioned, you're still working on the how, and my question is
Yes.
basically related to that. So I guess given two sort of facts. First, that of course, regardless of the context of the touch, MRGB neurons are going to be activated given a certain force. And then second, that even in the social context, there's going to be many neurons that are activated. Again, if the force is, light, basically. So given these two, facts, how are you thinking about how this social or stress-related, basically, modality is maintained and achieved? How is this specificity achieved with regards to the MRGB neurons?
So maybe I'll just restate your question so that I understand it. Are you saying that because the neurons can be activated by multiple stimuli, how is it that just maybe can you restate your question so that
Yeah,
I
sorry,
don't
it's
start
a little bit
rambling?
convoluted. Yeah. So basically, it's exactly like you said, right? So like these neurons can be activated by a variety of stimuli, regardless
Sure.
of like whether the touch is social.
Uh,
At the same time, even in the context of social touch, other neurons can be activated.
yes,
So
yes.
then how is this how is this specificity, do you think, is achieved?
Yeah, it's a good question. I mean, I guess I can speculate. I mean, if it didn't work, right, then I think everything you're saying is like valid. Maybe it's like, well, there as you get at, right, if you just do a social touch behavior, there are going to be lots of populations that are activated beyond this one. So just stimulating this small, relatively small one shouldn't do anything, or if they can be activated by other stimuli besides this social one, why would this like pathway have this like strong response? And I think if we didn't get a positive result, we would just stop, right? Because of the things you mentioned, right? But the fact that it does appear to be working perhaps could be telling us that, as Francis just mentioned, that this pathway, maybe others, a predominant function could be like, engaging in the stress relief. Maybe that's the kind of best way to answer it.
Got it. Yeah, I mean the results are very striking. Yeah, that's why I
Yeah,
was wondering.
yeah.
Ishmael, just one final comment I meant to mention. Is that vitamin T analogy, this dose response curve. You seem to need very little touch in order to get a massive effect. Like I
Yeah.
said, the rats had 3 or 4 minutes a day. The kids had 4 or 5 minutes a day, and that was it. Yeah,
Yep.
nothing else going on, no therapists, no instructions, just that. So,
Yep.
if your mouse models, can we get at the dose response curve, how much it takes?
That's a
Yeah,
great question. Yeah. Yeah. And we've
that
again we
matters for your therapy translational ideas.
yeah, I think we will need to do this, and one of our NIH grants, a reviewer suggested we have to do, and the light makes it amenable to do these dose response curves. We've done, just as you mentioned, we've done 3 to 4 minutes of stimulation. That seems to be sufficient, but um, we should look at 30 seconds, a minute, 10 minutes, and kind of get at exactly, which the point you're raising. We haven't done the dose response curve. We just, kind of started based on the literature, your work and others set off one time and went there, but it's a great suggestion. We need to do it.
It will stop the therapist charging lots of money for 2 hours of therapeutic touch, where 2 minutes will do.
Yeah, yeah, yeah.
Well, and that question, it's actually a really important question. Just if you look at, for example, the history of the placebo, the dose response of the placebo is so key in getting the
Hmm.
placebo into people's minds.
Hmm.
Like if you can titrate it up or down, such essential. All right, let's give Ish another round of applause. Thank you so much
Thanks for having me. Yeah, great
in
questions and comments.
today.
Thank you all.
And this is recorded. We'll post it on our YouTube channel. Thank you everybody again for attending. As always, the U24 Network. We're here, the PIs. Go visit our website, follow us on Twitter and Blue Social. And so thank you everybody. Have a nice day.
All right, take care. Bye-bye.
Thank you.